All right. All right, everybody. Thanks for logging on today. Another good, I should say, we have a very interesting topic to talk about today, something that I think a lot of us, as I mentioned before, don't know enough about. And I definitely don't know much about Danon disease. And I'm really excited to get to learn from Dr. Adler today. But before we do that, let me go ahead and introduce him. So Eric Adler is a professor of medicine at the University of California at San Diego. He earned his medical degree at Boston University School of Medicine. He completed his internship and residency at the University of Washington, and then a cardiology fellowship at Mount Sinai School of Medicine, where, as you heard, he worked with our beloved Dan Jacoby. And now he's the medical director of the Heart Transplant Program at the University of California at San Diego. Dr. Adler's research is focused on the study and treatment of cardiomyopathies, and recently his work has led to the development of a novel gene therapy for Danon disease, which is a rare cardiovascular disorder associated with severe hypertrophic cardiomyopathy. This treatment is currently being evaluated in a phase 1 trial. Dr. Adler also performs clinical research in heart failure and publishes on a variety of topics, including cardiac transplant, mechanical circulatory support, palliative care, and machine learning. So everybody, thanks for joining us. And Dr. Adler, please take us away. You're muted.
Oops. Thanks for the introduction. It's really lovely to get to speak with all of you. Hopefully, we'll do this in person one day. We'll get to see each other at meetings or something, though my enthusiasm, like yours, probably has diminished a little bit for resuming in-person activities. I'm going to talk to you today about a research program that I've started now almost 11 years ago that really started at the bedside. Here's a picture that I think as many of the advanced heart failure physicians are on can relate to. This is a picture from a patient. Actually, this is a patient with peripartum cardiomyopathy whose husband was a cartoonist, and on the whiteboard in the room while she was undergoing her heart transplant, he drew this. So when she came back to the room after surgery, we all found this kind of mural, and so to preserve it for posterity, I took a photo. But I always think it's funny that the surgeons are always the cool guys in all these pictures, and the cardiologists are always the ones with the clipboard and kind of looking geeky. But I guess we can't shake that. But anyways, so let's see. Here are my disclosures. Relevant to this is I'm a shareholder in Rocket Pharmaceuticals, so I'll be talking about some of the work I'm doing with Rocket. So this is a patient I saw when I was at Oregon Health Sciences as a early heart failure attending, so just about a year into my after fellowship. And he was 32 years old, and he had a history of heart failure and Wolff-Parkinson-White syndrome, and he was hospitalized for end-stage heart failure. This is at the time where the HeartMate 2 had just been out for like a year or so, and he was going into cardiogenic shock, and we didn't know him very well, and so we rapidly put in an LVAD. He was felt to have familial cardiomyopathy. We didn't have a ton of family history on him, but it was kind of one of these things where we didn't really have a good explanation. But post-operatively, as he was recovering from his LVAD, he actually was not thriving, and it was clear he had this kind of profound skeletal myopathy, a fact that we didn't pick up on when he first presented, because he presented pretty rapidly in shock. And we did a skeletal muscle biopsy. Here's some of the data that we gleaned from the patient. One, as we were rushing into his LVAD, we probably could have looked a little bit closer at this unusual echo, because his heart was enormous. I think it was 7.5 centimeters or something. I could be wrong, but his walls were quite thick, so it was a little bit unusual to be both dilated and hypertrophied, have hypertrophy of the right ventricle and the left ventricle. So that probably should have raised alarm bells that it didn't at the time. Then his skeletal muscle biopsy were notable on multiple different stains for the presence of intracellular vacuoles, which you can see here, here in red, and acid-fast stains. And then you can see also in electron microscopy, we can see actually mitochondria in a lysosome here. And this is double membrane bound autophagosome. So unusual looking electron microscopy. He was diagnosed by pathology with Pompey's disease, and we were starting enzymatic therapy for his Pompey's presumed Pompey syndrome. But I had a very motivated physician scientist in my lab that was also rounding with me, and she was really fascinated by this case. She was working in the lab, doing work on pluripotent stem cells, and I'll get more into that in a bit. And she decided to do a skin biopsy on the patient as part of a protocol we had to help phenotype patients better. And then, she had done some reading. She said, there's no way this was Pompey's, because adult onset Pompey's doesn't present primarily with cardiomyopathy. It didn't make any sense to her. So she sequenced for a gene called LAMP. So she's doing this all in the lab, aggressively moving. To be honest, she was supposed to be working on long QT syndrome research, but she shifted gears. She thought this was so interesting. So she pushed ahead. She sequenced him and found he in fact had a 2 base pair insertion into the LAMP2 gene, which led to a premature stop codon in the gene called LAMP. And I'll get into what LAMP does, but recognize here that in the patient's fibroblasts that she had now cultured in our lab, he didn't express any of this gene called LAMP, because there's no fluorescence here, where in a wild type you can see in this perinuclear fashion lots and lots of LAMP. So this and then Western blotting confirmed he didn't express LAMP. So this patient was basically a walking knockout. He was not, because of the premature stop codon that's relatively early, he was not making any detectable levels of the LAMP protein, or as far as we could tell at the time. So this was consistent with a different diagnosis, consistent with a diagnosis of, I should take it back. So that's one of the things I wanted to also point out about this, is that she was actually also rounding at the time with the skin biotome, which you won't see every day, cardiologists rounding with a skin biotome, but that's because she was in fact thinking about, could we make pluripotent stem cells from some of our inherited cardiomyopathy patients? Could we better understand the pathobiology of disease by biopsying patients? So I thought that was interesting. So it turns out she made the diagnosis of Danon disease in the patient. Fortunately, the patient expired, but we now had his cells growing in the lab, and it's a to be continued story. But just to give you some background on Danon, and frankly, 12 years ago, I didn't know much about Danon, and most people didn't really talk about Danon. But it turns out it's a highly penetrant, extremely morbid, X-linked, multisystemic disorder. So in boys, and this is a family that obviously allows me to share their pictures, presents with profound muscle weakness and hypertrophic cardiomyopathy that almost uniformly progresses to end-stage heart failure. Patients typically die in the 2nd or 3rd decade of life without heart transplantation. And as of today, there's no FDA-approved therapies for specific treatment of this disease. And you can see the echoes also show this severe cardiac hypertrophy, cardiomegaly at an early age. And the skeletal muscle weakness can be variable. So, I have patients that were playing tennis. I have other patients that rapidly progress. The cardiomyopathy tends to be the predominant symptom and is the more severe symptom. Here's an echo from a Danon patient, and obviously the takeaway is you can just see massive hypertrophy, biventricular hypertrophy, septums that can range on the 4 centimeter size or larger sometimes. So one of the clues that Danon should be in the differential is just the severity of the cardiac hypertrophy. In fact, there's a story that one of the first Danon hearts that was undergoing an autopsy, the pathologist couldn't even get it out of the chest using a normal dissection technique because it was so big to get out via sternotomy. So Danon is classified as a primary cardiomyopathy. I like to remind house staff, fellows, and even some of my colleagues not to try to move away from this term non-ischemic cardiomyopathy, because I think it's just a non-specific term, same as idiopathic cardiomyopathy, that we should be trying to better identify the phenotype and endotype of our patients. And in this case, we can see the various identify. This is from a paper from Barry Marin. This is way back in 2006, but you can see the various forms of genetic cardiomyopathy, whether it's hypertrophic cardiomyopathy, ARVC, left ventricular noncompaction, glycogen storage diseases, et cetera, mitochondrial myopathies, ion channel disorders. So, whereas mixed cardiomyopathy and acquired cardiomyopathy. So Danon falls in here. Some people will also call it HCM phenocopy. That's because it appears like sarcomeric hypertrophic cardiomyopathy, but some would argue it's not a sarcomeric hypertrophic cardiomyopathy and should not be referred to as such. So there's a lot of confusing semantics around Danon. It's similar to another disease called another mutation, PRKAG2, which also can affect, cause severe cardiac hypertrophy, and is also associated with Wolff-Parkinson-White, so remains in the differential diagnosis of cases like this. Danon disease is clearly a rare disease, and I remember when I first was starting to do research in Danon, many told me this was a fool's errand, that it was such a rare disease that no one would be particularly interested in the disease. A reminder that rare diseases are defined as by having less than 200,000 patients with the disease. It turns out there's at least 7,000 rare diseases. So in total, 25 million Americans have rare disease. And so collectively, rare diseases are common. And the struggle with patients with rare diseases is that no pun intended, they rarely can find physicians that have expertise in these diseases. They're complex, multiple organs are involved. There are usually, as in this case, but there's delay, the delay in diagnosis is on average around 4 years before diagnosis is made in a patient with a rare disease. And it's difficult to find specialists. And in hindsight now, I see this as a compelling reason to actually focus on rare diseases, or to focus on a rare disease in someone. It actually worked out very well at that stage in my career, because there wasn't a lot of other competitive interest in studying disease when it came to grant funding and things like that. And there was a lot of space to operate. More importantly, these patients have this very morbid disease and were being ignored. So I found it very personally gratifying to become a reason to come to the lab, to come to work, to be passionate every day. So I gained a lot of empathy for people that have rare diseases. And then finally, it kind of becomes like a reason to go to quaternary care centers. Like, what do we do that the community doesn't do? Why should patients, when they can go to 10 different private cardiologists in your area, and go to a quaternary care center? This is something we're going to do better. Because we can work together. I think we can have multiple kind of very highly specialized physicians under the same roof, which these patients often need. B, we have these research interests. For all these reasons, I think rare diseases are particularly well treated at quaternary care centers. So anyways, with that being said, so this is kind of a summary of the options. The other reason, which I forgot to mention, I'm just reminded here, why it's interesting to study rare disease. It's usually genetic, but not always, but they usually have an underlying specific root cause, a molecular root cause that we can identify. And now with all of the new tools that we have around as we saw in the paper just yesterday with this pig heart being transplanted, but we can change genes, add genes, delete genes, etc. We can move closer to treating the root cause of these disease and develop kind of a personalized approach to these patients, a specific approach to their disease, as opposed to the broad approaches that we use generally in heart failure with GDMT. We can really be focused on the underlying root cause. So what's the root cause of Danon? So it's due to mutations in LAMP. And here is LAMP protein here. It has this small cytoplasmic tail. Here's a lysosome. This is where LAMP is found, because lysosomal associated membrane-bound protein. And then inside the lysosome is this protein, the luminal side of the protein, which, to be honest, we don't know a ton about the function of the majority of the protein. What we do know is that, and I'll get into it, is that there's 3 isoforms of LAMP, and they differ from this tail here. And this changes in this tail, the cytoplasmic tail, seem to dictate the function of the protein. And then we also know that the most commonly expressed isoform of LAMP in the heart is LAMP2B. So that's the one we wanted to focus on. So that was the most abundant isoform. If there's A, B, and C, B was the one that's 10x expressed in the heart. And then loss of LAMP2 profoundly affects a process called autophagic flux, and I'll get into that. So here are the mutations that are found. I think now there's close to 90 mutations identified in the LAMP protein. Many of them are early in the protein, leading to loss of function. And in fact, there are very few mutations in this disease that are not associated with loss of function. Some of these in the isoforms, like there's mutations in LAMP2B specifically, which cause cardiomyopathy, again, which further supports the pivotal role of this specific isoform in heart failure in Danon patients. So Danon is critical for autophagy. This LAMP protein is critical for autophagy. Autophagy is a very trendy topic these days. I didn't realize it when I was starting to study it. It's trendy because it's, Dr. Osumi won the Nobel Prize for it in 2016, but also you may have a lot of patients, or you may yourself be interested in this kind of fasting diet. And the fasting diet, the root kind of basis for doing fasting diets is that you start doing autophagy. So anytime you're starving for more than 4 hours, you have vesicles that form that are called autophagosomes that go around your cell and scoop up mitochondria, other proteins. They can scoop up RNA, they can scoop up viruses, all kinds of processes. This is a ubiquitous process. It occurs in pretty much every multicellular organism from yeast on, do some kind of autophagy. Then after these autophagosomes fill up with this junk particles, they bind with lysosomes, and lysosomes have a low pH and have hydrolysis in them, which digest the content of the autophagosomes. That produces single chain and branch chain amino acids, which can then be used for energy. So it's a process to help get rid of the waste in the cell, specifically, and it seems importantly, to digest dysfunctional mitochondria, but B, also to generate energy. So it's a recycling system for when you're starving and to still generate energy. And you can imagine the heart is working 24 hours a day, so it's particularly dependent on autophagy. And Danon proves that, because Danon mostly affects the heart. And so, what does LAMP do here? Oh, I should say also, one of the interesting things about autophagy and LAMP specifically was, this paper that Saftig, Paul Saftig wrote in, or there's an editorial that Paul Saftig wrote in about 10 years ago, showing that when you overexpress LAMP in a normal mouse, it can actually live longer. So just like when you starve a mouse, it can live longer. If you overexpress LAMP, it can live longer. Just like if you give rapamycin to a mouse, and it lives longer. And remember, or I don't know if you remember, but rapamycin, one of the key things it does in a cellular level, even at low doses, it induces autophagy. So there seems to be some signals here that suggest that autophagy and this protein lamp may have a direct impact on lifespan. What does LAMP do to cells specifically? Well, LAMP binds to, sits on the lysosome, and one of the functions we think that it does, and there's multiple, but the function that seems most prevalent in the heart is to allow for fusion of this lysosome and autophagosome, or at least be critical to that process. And there's still a lot of work being done in this area, and I'll review some of it shortly. But you can see in Danon disease, they don't express any LAMP. And so you're not able to have this fusion. And one of the things I mentioned here is this protein LC32. That's a protein that sits on the autophagosome that builds up when you can't do autophagy. And it's relevant because it's a marker that we use to tell if autophagy is working in a cell. So Danon patients don't express LAMP. So this process of fusion between autophagosome and lysosome is greatly impaired, and LC32 actually builds up in the cell. Shifting gears a little bit, how do we study these rare diseases? Well, one way that we study them is using pluripotent stem cells, or induced pluripotent stem cells, or IPS. This was back from when I was doing postdoc work at Mount Sinai, a paper that I was part of that Sonia Carval wrote, looking at a different rare disease called Leopard syndrome, where just like Dr. Clegg, when she took a biopsy of this patient, we took a biopsy of a patient with Leopard syndrome, used a number of inducible factors developed by Shinya, or who were first described by Shinya Yamanaka, and we could make stem cells in the dish that mimic the disease from the patient. So this is a pivotal finding, that in this case, when we took skin cells from a patient that had hypertrophic cardiomyopathy, convinced them to be cardiac cells through this long process, they were hypertrophied. So they retain key features. So this allows us now to study diseases in a dish in a way we never thought possible, human diseases, because we're always using mouse models and everything like this. So it's a big thing moving forward. This process of using pluripotent stem cells to study human cardiomyopathies has exploded in the last, and it may seem kind of behind the scenes for most clinical folks. Most of the attention here at this time was focused on, can we use these stem cells to regrow hearts and inject them into hearts, or inject parts of them into heart? That is still ongoing. That journey, it's still uncertain if that's going to work. But what is clear, what's working, is that we can study diseases, long QT, Leopard, HCM, DCM, catacol, CPVT, etc., using this stem cell model system. So one of the first things we did was we made stem cells from this patient, and we just started looking at calcium handling, at all kinds of features of the cells. This is how we look. I think this is really cool. This is how we look at calcium handling in the dish. So we actually, we use a fluorescence dye in which calcium is green, and we can basically generate EKGs from each individual one of these stem cells that we've made from the patient. So this is from a Danon cell, and we can then create action potentials from thousands and thousands of cells in a very short period of time. So you can imagine doing clinical trials, we can test drugs on thousands of cells and look at the effects of drugs on action potentials using this system. And we did it with the Danon cells and multiple, we analyzed these Danon pluripotent stem cells in multiple ways. I'm going to fly through some of this because I want to get to more of the clinical stuff. So bear with me, but we made the stem cells from the Danon patient who had now expired. We saw that these cells had impaired autophagy. We saw that they actually matched the patients where they had these lysosomes and autophagosomes, just like that first patient did. So now we had a disease that where what we were seeing in the dish really matched what we were seeing with patients. We then went on to describe these cells and show that the cells had critical features that were consistent with cardiomyopathy, lots of ANP and BNP, impaired calcium transients, hypertrophy. So features consistent with cardiomyopathy. They were abnormal looking compared to a wild type cell. We then looked at mitochondrial function, and across it, we did RNA-seq on these cells and saw that they expressed a lot of abnormal genes that were consistent with mitochondrial dysfunction. So that gave us some clear evidence that Autophagy, as I had mentioned, it's critical for digesting mitochondria that are abnormal. These cells don't do that very well. And as a consequence of that, their mitochondria behave. And that might be one of the root causes of disease in these patients. And that's consistent with a lot of other inherited and non-inherited cardiomyopathies, where we see this kind of final pathway is mitochondrial impairment. Here's some more evidence that mitochondria are impaired. We went on to then move on to get mice with Danon disease, and again show that they're very similar. This is working with Paul Saftig in Germany, that they not only expressed a lot of these pathways that were abnormal, but they also had impaired contractile function. So this is all laying the groundwork for creating these model systems is critical for developing new therapies. And you could ask yourselves, why do we have all these new therapies for HCM and for Danon and all these things, and we don't have many new therapies for sarcoid? Simple thing. One of the main reasons is we don't have good model systems to study sarcoid, right? We don't really have good animal models for sarcoid. We do have good animal models for some of these other diseases where you're seeing advancement. And as a consequence of that, you see we can really move forward with new therapeutics, but you can't move forward if you don't have model systems. So it's so important. But here now we had a stem cell model system and now a mouse model system to work with. We went on, and I'm not going to belabor this, but we made our cells in mice so we could actually track autophagy in real time based on fluorescence. This is working with Joe Hill at UT Southwestern, who became a close mentor and partner for me on this project. And we can now have this little factory where we could tell if cells and mice were behaving or autophagy was impaired or not by using these different reporter systems. And I'm going to skip through some of this. We showed that the mice had abnormal mitochondria. I'm going to skip through this because, tailoring this to this audience specific. But the bottom line here is we were at this kind of pivotal point in this journey. This is about, now we're talking about 5-6 years ago. We were making a lot of basic insights into how Danon disease impaired mitochondrial function, and we could get, writing grants, kind of more basic grants. But at the same time, patients were actually calling me and finding me on Facebook and emailing me and showing up to San Diego saying, my son is dying with this disease. There was me and this other gentleman, Matt Taylor, who's a close friend and colleague of me, were essentially like the only people at the time really actively focused on Danon. And they were saying, can you develop treatments? The clock is ticking. And at this point, we decided to make a little bit of a pivot. So I use this analogy of the truffle pig versus the racehorse. So the truffle pig is like basic science here. It's actually very important, and a truffle pig is way more valuable than a racehorse. It's finding these truffles every day, and it's not as glamorous a job. It's hard work. You sniff out and try to find these truffles, but you have a long lifespan. Truffle pigs are actually way more valuable than racehorses. Racehorses, and I think of the truffle pigs analogy as being discovery science, these basic root studies. Lots of work, laborious, but over a whole career, you can make major insights and be very impactful. Translational science is like the racehorse. It's a sprint. If you find something and you're lucky, it can be very beneficial. But most racehorses, most translational science endeavors will fail. And they're shorts, they move fast because the clock is ticking in this case. But if you find something good, it can be incredibly impactful. And this is true in this case. We decided that, we knew the root cause of disease was the loss of LAMP. And as opposed to just focusing on understanding basic insights into Danon disease, we wanted to develop therapies. So we started doing drug screens. So the first thing we did is we took our stem cells and we put them through these high throughput microscopes with every known FDA compound. And we looked for compounds that affected the cells. Remember, I showed you that green, yellow, red system. So we could tell if cells were healthy or not. And then we could put literally overnight every FDA approved drug, thinking that we could repurpose. And we did this at the California Institute of Regenerative Medicine, trying to show that we could use stem cells to identify and repurpose drugs that existed. So we did this screen. we looked for cells that gave off a certain fluorescence that could tell us a signal that they would be helpful. And the screens look like this. Every little line is a compound. And we found a lot of compounds that made cells worse. So for example, a lot of the anthracyclines made cells worse. A lot of other chloroquine made cells worse. But what was interesting is that there weren't a lot of compounds that made things better, and nothing worked better than we had just, quickly in the lab and didn't put much thought into it, made a viral vector overexpressing LAMP as our control. And that viral vector, and we were just using a lentivirus at the time, completely reversed the process. So the vacuoles dropped significantly. So at first we were kind of hanging our heads low, like, oh, we're not going to find any drugs to treat these patients. We kind of had this aha moment. And this was, again, a theme here is clinical cardiologists who are also doing research. This is Brad Nelson, who was working in my lab in the corner, and Shireen Hashem, who's now a clinical pathologist, both interested in basic science, but working in the lab. And we were sitting around talking. We were doing clinical trials on gene therapy. If you remember the Cupid trial and other things. Said, well, why don't we just put LAMP, this protein, into AAV9, which had been shown in other studies to be highly effective at infecting the heart. So we made a gene therapy using AAV9, a virus that had now been used, was being used to treat Duchenne's disease, to treat some other skeletal myopathies, now to treat cardiomyopathy, knowing from other research done that AAV9 is very efficient at infecting the heart. So we made a construct, I should say, in which we put LAMP2B, which, remember I said was the protein most highly expressed in the heart, into AAV, and it was expressed by a very potent promoter. And we saw right away, this was like an aha moment for us, that we could in fact rescue. We could either express LAMP2A or LAMP2B. We tried both in the heart very efficiently. So we were now giddy. We were like, wow, we can get this protein that was missing into the heart of these mice. And we moved pretty quickly. This is 2 years worth of work that Maria Monso did in the lab, where we infected a whole bunch of mice that had this model of Danon disease with this vector that we had made. At this point now, we were partnering with a company called Rocket Pharmaceuticals, who's interested in licensing this product. And we overexpressed LAMP in the mice, and basically mice stopped dying. Their hearts, here's impaired contractility based on we actually were doing invasive hemodynamics and looking at cardiac output, or in this case, DPDT max. And with virus, we could almost completely restore normal cardiac systolic and diastolic function. And then what was most exciting for us was that on electron microscopy, basically you saw resolution of these kind of abnormal mitochondria and vacuoles in the mice. So, in the last 10 minutes or so, I'll tell you what we've done since then. And actually one of the problems that we faced, and what we didn't realize, was that the truth is, even if you have a potential cure for a therapy, if you do not know the natural history of the disease well, you cannot even start a trial. The FDA won't let you start a trial because they said, what endpoints are you going to study? How can you prove that it's safe and fair and ethical to infect patients with a virus if you haven't really shown me this disease is so bad? I said, oh no, it's terrible. Where's the data? Well, there were no really good published. There were some, but nothing great that the FDA would accept natural history studies. So we were kind of at the sticking point where we couldn't proceed with the trial. So this is work that Michela Brambati did while she was in my lab. She's an Italian cardiologist. We quickly did a structured meta-analysis of Danon disease. So this, for people interested in research, is a great place to start. Michela had done one on eosinophilic myocarditis. But the principle here is that you take every case report, you deconvolute it, you put it into a new database and you treat that as a natural history study. And that's a first quick, it's obviously not as good as a true natural history study, but gives you a first glimpse in the study. So she deconvoluted these 750 papers, found the ones that were good, and we get this kind of picture of Danon disease, at least enough to show the FDA that we could proceed with phase 1 studies, showing that, you know, most boys die or needed VAD or transplant by age 30, and that even women, and this was kind of surprising, and this is a focus of our research, were dying maybe later, but we're dying from Danon. And then using this meta-analysis system, our meta-analysis that we did, we created this kind of construct. This is all, you know, with obviously significant caveats here, but showing that the when the, you know, onset of hypertrophic cardiomyopathy occurred, cognitive impairment was more common than we thought, myopathy, when people got defibrillators and stuff, all from a meta-analysis. So you're a fellow, you have a good idea, rare disease you want to study or a rare condition you want to study, this is a great first place to start. And you don't need a huge database. You can build a database from existing papers. So, but we had no patients. So now we had this disease, highly we're interested in, very morbid, but the patients are all over the world. So what did we do? We went to IRB, we said, can we try to find patients on Facebook? So we put a calling out there on Facebook. Hello, like, is there anyone with Danon disease? We'd love to connect. And it turned out there was a Danon disease support group. And it turned out once people with Danon disease heard that someone actually cared and was studying it, this just blew up. And we had patients joining the group. We found patients from all over the world, certainly all over the United States. This map is outdated. We have patients in South America. I was in Brazil on 60 Minutes of Brazil talking about Danon because they had a bunch of Danon in Brazil, Australia, Canada, Europe, etc. Now all willing and able to participate in natural history studies. And so we have patients from around the US now participating. And now what we had gotten funding for is a prospective study and a cross-sectional study of Danon disease. So moving from this meta-analysis, an actual study of Danon, and we found that the two things matched up pretty well. When boys got heart failure matched up to when we thought, we found some interesting things that boys are more likely to get hypertrophic cardiomyopathy, women are more likely to get a dilated cardiomyopathy. Lots of interesting things in terms of the prevalence of disease that we learned from doing the natural history study of Danon. We learned a lot about MRI of Danon, so then we went on to do this prospective study of magnetic resonance in Danon, and this is work that Marcia Rigoli did when she was doing her postdoc at UCSD, where we found specific interesting uptake patterns of gadolinium in which the septum is spared. So unusual patterns, impaired strain. So characteristic MRI findings that might help you when you MRI a HCM patient or a potential amyloid patient that might say, oh no, this looks more like Danon disease. And that's important. If you remember, up to 20% of patients that getting myectomies at Cleveland Clinic, for example, this is a paper they published, didn't actually have sarcomeric hypertrophic cardiomyopathy. They had other diseases like Fabry's Danon, these phenocopies, amyloid, etc. So looking for clues that this is not just plain old sarcomeric hypertrophic cardiomyopathy is very important when you're considering therapies for these patients, because they're going to behave very differently. Here again, some characteristic findings. This was a paper from Jack Imaging that we did showing the sparing of the septum and gadolinium in these patients. It's not 100% specific, but something that might lead to thinking about Danon. This is work that Quan Bui is now doing in the lab, looking at strain patterns of Danon, which are actually quite similar to amyloid. So again, this kind of cherry on the top pattern, very abnormal strain. You see a 20-year-old, it's very unlikely to be amyloid. So you see a 20-year-old with hypertrophy and cherry on the top strain pattern, you may want to think about Danon. We then started shifting a little bit, and this is the last few slides, into even, as we do these natural history studies and as we prepare for a clinical trial with the gene therapy we developed in the lab, what endpoints can we use? What surrogate endpoints can we use to predict mortality? Knowing that if we pick any group of patients within 1 year, it's a rare disease, we're not going to find patients that predictably go on to die within a year. So we're going to need some surrogate endpoints. And some of these patients don't have impaired exercise tolerance yet. So we need to have surrogate endpoints to show, and this is again mandated by the FDA, makes a whole lot of sense. And it's very relevant to amyloid trials, to other trials. What can we use to predict the efficacy of drugs short of 6-minute walk test, short of, CPET? This is particularly relevant in Danon because our patients with Danon also have skeletal myopathy. So 6-minute walks and CPETs might not be indicative of therapeutic response. It's imperative in a disease like Frederick's, in which again, 6-minute walks and CPETs might not be your best test because they get myopathies. What Quan finds here is that strain more than ejection fraction predicts outcomes. So as opposed to, in heart failure, we're always, as more guidelines come up, we subset our patients into HFrEF, HF middle range EF, preserved EF, all of these things. When we're really trying to predict mortality, strain actually, at least in Danon, and we suspect this would be true for other, and we're doing work on other sarcomeric diseases, that strain ends up being much more relevant than ejection fraction. And you can see the patients with the lowest strain were the ones that were going on to transplant. And it wasn't until patients really dropped their EF that their mortality was bad, but you missed a bunch of patients in here that had preserved EF, but that were doing quite poorly. We did some work on electrophysiologic characteristics of Danon. So this is again from our natural history study, just showing that Wolff-Parkinson White might be a prognostic indicator in Danon, that if you had WP double, you're more likely to die of Danon. But one of the things in terms of medical mythology is that the majority of Danon patients don't have WPW. So it's not a sensitive finding. Only 33% of Danon patients actually have WPW. But if you have Danon and WPW, you're more likely to die. And then finally, a lot of these patients, especially in the community, end up going for ablations, but they turned out to have fascicular pathways and AV pathways. They are very hard to ablate. So we don't recommend ablation in most of these patients. It's almost always unsuccessful because they have so many pathways. It's not your traditional WPW kind of pathway. We also learned that even though in this small registry, many of our patients had ICDs, we didn't have a single patient who actually required his ICD at this point. So when to put an ICD in a Danon patient is still a matter of debate. And obviously there's not really good guidelines. So you're kind of stuck. We're using the amount of fibrosis and delayed gadolinium as an indicator for a defibrillator, and obviously drop an EF as an indicator for defibrillator as per HCM guidelines, but we're not really sure. Clearly, we don't want to be wrong, so we err on the side of defibrillators, but we don't really know. So we've started most excitingly for me is we actually started the trial. So we started the trial in 2019 due to conflict of interest and other issues. Barry Greenberg, my partner up here and mentor, is the PI of the trial, and that's been ongoing in enrollment, and hopefully we'll move to pivotal from out of phase 1 into phase 2 trial shortly. And here's some, if you have Danon patients who might be interested, here the trials enrolling actually at CHOP in Denver and in UCSD for now. So just wrapping up here, you can see this approach of understanding the root cause of disease and developing gene replacement is feasible. Danon may be one of the first examples of doing this for a genetic cardiomyopathy, but there's a whole lot of other genetic cardiomyopathies out there. And it turns out since we've been doing this Danon work, there's been a whole lot of companies popping up doing work in this area. So there's currently in development right now, there's a gene therapy for myosin heavy 7, for myosin binding protein 3, for TNNI3, for lamin, for obviously Duchenne's, for PKPD2, for DSP, for ryanodine. So we're seeing what could be this paradigm shift, where as opposed we're treating all these patients with genetic cardiomyopathy essentially the same, that we may, in the era of gene replacement and even gene repair, develop targeted therapies for these rare diseases that treat the underlying root cause and hopefully provide us cure. To support this work, now we've started a center here for anyone interested called the Strauss-Wilson Center for Cardiomyopathy, whose focus is these genetic cardiomyopathies and developing new therapies for them. So developing translational therapies for cardiomyopathy and doing the natural history studies, doing the basic science work, doing large animal work, doing pig models of disease. So lots of exciting things to study these causes. So that's it. In conclusion, these rare diseases are understood but are morbid, provide mechanistic insights, and collectively affect many people. Stem cells in this case, maybe the jury's out on whether they're a good therapy, but they're certainly an excellent model system for study of disease. Again, structured reviews that we did here ended up being very impactful in our work and relatively easy to do. If you're interested in research questions. And finally, the jury's still out, but what we used to call idiopathic cardiomyopathy may in fact be quite treatable with gene therapy. And finally, this all started at the bedside. And I think I have an audience that feels the same way, because I know many of you, but our patients are our best inspiration. In this case, I was inspired by our patients, and your inspirations can directly impact your patients. So it's a full circle, and it's a wonderful place. We have all these challenging conditions and the burnout that we go through and the amount of bureaucracy that we all deal with that we're feeling. And I can tell you that being engaged and interacting with our patients and being inspired by our patients like this is a great treatment for those feelings of burnout. And then finally, the best parts of your career, and in this case for me, have been these many friends that I've made doing this kinds of research. And in fact, the friends I've made may be the single best part of all the research things I've done. So here are some Danon patients that come to the lab from all over, that make these journeys. Many of them were coming before there was a therapy, just willing to help, and incredibly inspiring for our group. No one complains about how hard they work when they see these patients. And so I'm very thankful for getting to work with them and lucky to get to take care of them. Here are members of the lab and my family that have all worked hard over the last 10 years to put this story together. Here's our transplant team and some tattoos that our transplant team made with our SRTR outcomes. And here's our funding. So thanks so much for inviting me here. I'm really honored to be with. I'm so impressed by all the things that you guys have been doing in your section and in your division in the last decade or so or longer. So thanks for inviting me to come to speak. I'm happy to answer any questions.
Thanks so much, Dr. Adler. That was, obviously that was a, very inspiring talk, and just the things that, the opportunities that you're opening up for so many, patients with inherited cardiomyopathies. It's going to be really exciting to see where this goes in the decades to come. Does anybody out there in the group have a question that they'd like to start off with Dr. Adler?
Hey, this is Dan. I have a quick question. Well, first of all, Eric, like so amazing, just absolutely incredible, like kind of speechless with regards to all this amazing work that you've done. Great talk. I was just kind of curious, like, how did you fund different things along the way? So, like, you had some K money, you had some foundation money. Like, how did you figure out how to pay for your postdocs and run your lab and all that stuff?
Yeah, I think this is a really good question. It's funny, I didn't put it in this version of the slide, but I mean this version of the talk, but in other versions of this talk, I keep putting in these rejection letters I got from the NIH. Like every 10 slides, I put in my NIH review committee study response to remind people that don't be despondent. And my responses from the NIH were interesting over the last decade, because at the beginning, the responses were like, oh, this is a really rare disease. This is never going to work. These approaches, are too translational and not basic, or too basic and not translational. And then over time, my latest response from the NIH was, you've already done this, so why should we fund you? So it went from like, this is never going to work, we're not going to fund you, to like, you've already done this, we're not going to fund you. So it was super frustrating. But I think it's important to think broadly and out of the box. We definitely had some NIH money, we had some AHA money, but not enough to support this work. We had some money from CIRM, from California Institute of Regenerative Medicine. So that was, the first few years I got to UCSD was funded by CIRM to do these screens. But to be honest, it was very, this is 5 years ago, venture capitalists came up to me, kind of cold called me, saying, we're starting a gene therapy company and we'd like to fund your work. And I thought it was like a prank. I was like, why is anyone calling? They looked me up. This guy, Rod Wong, looked me up and saying, we think this is really important, and we'd like to develop a rare disease company, basically around, whose foundation was this Danon project, or one of the foundations. And so, and then finally philanthropy. So there were patients that were also generous, that were suffering from this. So thinking beyond the NIH, I think is really, that's the bottom line. It's really important. Working with, there's a lot of, I think we're moving where industry is really interested in all these things, because things can translate so fast. So I think partnering with industry is not taboo. It's actually, if you really want to translate, if you're at that translational stage, it's been a great partnership for me, for sure.
Thanks.
Anybody else is welcome. I guess while if anybody else might be thinking of a question, some of the things I were thinking about during your talk, a little more clinically related, and just really because of my lack of experience of ever taking care of anybody with Danon. So I guess the first thing I was going to ask you, just in general, I mean, if you've gone through transplants with patients with Danon, what's their prognosis like after transplant? How do they tend to do?
Did I not? I think I skipped through I'm sorry, I skipped through a whole slide. Can you guys still see my slide deck?
No, I took you off the share screen. Yeah.
Let me,
But you still should have the ability to share.
Yeah.
I think. Hold on.
Yeah, I got it.
Okay.
So we just wrote a paper on this, and because in general there had been a consensus or a prevailing thought that patients with Danon didn't do well with transplant, and they actually do okay. This Kaplan-Meier curve is actually probably pretty similar, right, to what you would see, or if not as good or better than what you'd see in a non-Danon patient. Obviously, ideally, we wouldn't transplant these patients because they're young. So, anytime you're transplanting a 10-year-old or even a 20-year-old, we know long term it's not an ideal outcome given the longevity of the organs. But they generally did okay. I would prefer they're taken care of at centers that deal with a lot of genetic cardiomyopathies that are used to kind of harder, because sometimes they'll struggle and they're not necessarily the easiest cases to do. So I don't think they're the best patients for like small centers. But I think most, the larger centers, these patients are going to do well. And we've had patients do well that get transplanted. Their skeletal myopathy, they're, in theory, rapamycin should make them worse because it makes the mice worse. So we've been avoiding rapamycin, but we don't know that for sure. And now we're just starting to say, well, maybe if it's at low doses, it actually might be okay. But there's a lot of gray area here with transplant.
And then another question I had is just like kind of, again, clinically, if we're looking for patients or, we're presented with patients who are relatively young and they look like they may have hypertrophic cardiomyopathy, and thinking about phenocopies of hypertrophic cardiomyopathy, is there anything, I guess, outside of the heart disease that could clue us into a diagnosis like this? And then is really the only, I mean, I know you'd mentioned some of the non-invasive imaging studies, but really is the gold standard for diagnosis of the endomyocardial biopsy and getting a sample?
Well, I think, and I probably should include this in my talk more, but I think the standard is the genetic test in the right patient. And, you know, and I think Dan would appreciate this. He and I probably talked about it, is that there was a prevailing kind of sentiment, like, why bother to do genetic testing in cardiomyopathy? Like, you're going to do the same thing. Why bother doing tests? And even in the last year or two, I have colleagues in hypertrophic cardiomyopathies, like, what's the difference? This kind of nihilistic thought about why bother doing genetic testing at all? We're going to do the same thing. I think we're not. I think this is more reason than ever, besides the whole implications for families. So I think it's actually, I would argue, it's almost negligent not to refer your patients, your HCM or idiopathic patients for genetic testing, because you can save their kids' lives. And I've done that. And I've also missed it and had, you know, taken patients and had kids die on the soccer field while I was taking care of the patient in the office. I mean, just terrible things. So that's a reason by itself. But beyond that, I think anytime that you see any cardiomyopathy patient with systemic symptoms, like in Danon's case, it's, you know, vision problems, in not doing well at school, some skeletal myopathy, that's a sign of a rare disease. So anything that's not quite right outside the heart, that's usually a clue that you're dealing with some kind of genetic rare disease. And it could be Danon, it could be, you know, a whole host of, you know, other lysosomal storage diseases, whether it's Fabry's or, you know, Pompey's, etc. So that's a clue. Danon would be specifically skeletal myopathy, you know, some learning disability. Those would be bigger, and then some vision problems. And they have very specific visual things. But I think I get it. 10 years ago, my patients were getting these big bills for genetic testing, right? They were getting thousands of dollars in bills, and insurances weren't covering it. But now, you know, if it's not free, it's $200, something like that, for genetic testing. You know, not all of us, and you know, and I'm curious what Dan thinks. Not everyone agrees, but I think there's no reason not to genetically test patients right now.
Yeah, Eric, I mean, I'm totally on the same page with you. And I think our whole team has kind of come, we all kind of function together this way. When we see patients in the hospital, we get our genetic counselors to see them. And we've sort of been talking about instituting routine genetic testing for all non-ischemic cardiomyopathy patients that come to transplant. So I couldn't agree more. And it's very applicable. I want to also just thank you once again for coming. Mike Beasley and Tarek have set up these incredible lectures, and it's just such a privilege. And this one in particular for me is just so wonderful. And thank you so much. I have to jump off because we have our HCM clinical like myectomy meeting at 4.
Sure.
And I sent you an invite, so you should jump onto that call.
Awesome. Well, thanks, guys. Thanks so much for having me. I really appreciate the opportunity. I hope to see you guys in person soon.
Thanks, Dr. Adler. Thank you for taking the time.
All right. Take care.
Bye-bye.